In this unit, we're going to talk about cell structures and their functions. And you can see that there's a lot of information that we expect you to know. But the good news is all the information that is included in this column is material that you were already taught in pre-AP, or it's going to be review material from the biochemistry unit. For example, one thing you remember is that bacteria cells make their walls out of peptidoglycan. So you're going to see some information from last unit pop back up in this unit. There are a few additional details that we're going to teach you that are going to be new, but the majority is going to be review. Now, to see how much you do remember, you might hit pause and see if you can fill in these blanks of this second column right here. Eventually, though, at the very end of the notes, we're going to go back and we'll fill in all those blanks so you have the correct information. Now, we're going to go ahead and talk about cell parts that interact to produce proteins for the cell. Your typical AP question is not going to have you just tell me what is the function of the rough ER. You're more likely going to be asked, how does the function of the rough ER affect the Golgi or the plasma membrane? So you're going to have to really understand what each cell part does so you can tell me how it would interact or affect other cell parts of the cell. Now remember, we know that DNA codes for the proteins. So this process of making proteins starts with the copying of the DNA molecule into an mRNA molecule, and the mRNA molecule is then sent to a ribosome. But we need to make sure you understand that there's two different types of ribosomes. We have attached ribosomes, and I can tell that these ribosomes are attached because it's going to make a protein that it sends to the rough ER. But mRNA can also go to unattached ribosomes. Those are ribosomes that are floating around in the cytoplasm of the cell. Now regardless, ribosomes are going to make proteins. You can see that in this diagram here. There's an unattached ribosome, which is right there. It's reading the mRNA, so this would be mRNA, and it's putting amino acids in order to make a protein. So here's the protein. But one thing you need to be able to tell me is that unattached ribosomes are going to release those proteins into the cytoplasm of the cell. So unattached ribosomes are making proteins that are going to stay in the cytoplasm of the cell. Now, your attached ribosomes are ribosomes that are on the ER. So here's the ribosome, and here is the surface of the rough ER. And you can see that the ribosome is reading the mRNA molecule, and it's making the protein or the polypeptide. Now, you need to understand that this polypeptide can end up going to 3 different places in the cell. Some of these proteins that are made and put into the rough ER to be modified will end up in a lysosome. Some of these proteins might end up being released from the cell, and we also can say that these molecules are being secreted from the cell. And some of these proteins might end up being things like the transport proteins and the marker proteins that are embedded in the phospholipid bilayer of the cell. So again, to summarize, your attached ribosomes are going to end up making proteins that can be embedded in the membrane or released. It's also making proteins that can end up in lysosomes, whereas your unattached ribosomes are going to make proteins that are going to be simply released into the cytoplasm. One thing I want to mention is that many times students are saying that ribosomes, like you see here, are making, and they'll say that ribosomes are making ribosomes and putting the ribosomes in the rough ER to be modified. So watch out with your terms there. Ribosomes are making proteins, and then they're sending those proteins or stuffing those proteins into the lumen or the inside of the endoplasmic reticulum, and then the rough ER is going to modify those proteins and then send them on to the Golgi. Knowing this information about how proteins are produced should give you the ability to look at diagrams like this and understand what it's showing you. So here we have the nucleus, and you can tell that this is going to be the rough ER, and those little dots on the rough ER would be the ribosomes. So that would be a ribosome. And since you have a molecule inside of the rough ER, you can assume that is a protein. That protein was made by the ribosome. And you can see that that protein is eventually being sent to the Golgi. The Golgi is the post office of the cell. And it can ultimately tag the protein, which we see right here, to go to its final destination. And in this case, it's tagging it to be secreted, which is one possible destination for proteins that are made by attached ribosomes. Here we have, again, a different protein that is in the lumen of the rough ER. You can see that that protein is being sent to the Golgi. The Golgi is tagging it for its final destination, And in this case, it looks like that protein ended up being enclosed in a phospholipid bilayer because that protein is now inside of a lysosome. And we're going to see here that the Golgi is responsible for making lysosomes. So this little vesicle from the Golgi is going to break off and it's going to form a structure called a lysosome. When we look at this diagram over here, this is attempting to explain to you how proteins can be embedded in a phospholipid bilayer. So if you look at right here, there is a protein, and that protein would have been made by the ribosomes, and it is embedded in the phospholipid bilayer that makes up the rough ER. And so whenever this piece, this vesicle, breaks off of the rough ER, you can see that we have a protein that's embedded in the phospholipids, because this is really just a phospholipid bilayer there. So ultimately, that protein is going to be sent to the Golgi. Here it is here. The Golgi is tagging it for its final destination. And then when this vesicle fuses, it becomes a part of the phospholipid bilayer of the plasma membrane. And you can see now, we now have an embedded protein. And this could be a protein like a transport protein or a marker protein that we know are proteins that are embedded in the phospholipid bilayer of cells. So let's fill in some of these blanks here so that you know if you're trying to describe protein production, you know how to use these terms. So we have ribosomes on the surface of rough ER. They produce proteins that can be released into, now the inside of the rough ER is called the lumen. And I just want you to be familiar with this term. You could say released into the rough ER. The rough ER is going to modify those proteins. For example, it can attach on like carbohydrate chains. I'll show you an example of this on the next page. So these modified proteins are now going to be sent to the Golgi. Something I want you just to be familiar with, so it doesn't throw you off if you see this complex term, is the receiving part of the Golgi is called the cis phase, whereas the shipping center is actually called the trans phase. So if you want to put trans phase. But ultimately, the Golgi is going to tag it and send that protein to its final destination. It can be sent to the membrane where it can be secreted. That means released into the extracellular environment. That's one thing that can happen to that protein. It can be embedded as a membrane protein. And then also, some vesicles from the Golgi are going to end up forming lysosomes. And inside those lysosomes are those proteins that were made ultimately by that attached ribosome. Now, this diagram down here is just attempting to show you that the Golgi is responsible for the production of lysosomes. And also, just to give you another term, we usually show that inside the lysosomes there's these little molecules, and those would be the hydrolytic. You should know what I'm talking about when I say hydrolytic. These are enzymes that are going to assist in hydrolysis, the breaking down of molecules into their subunits. We learned that in the biochemistry unit. But these are called hydrolytic enzymes because we know that the purpose of a lysosome is to help break down molecules of the cell. And so it would contain hydrolytic enzymes that were made by attached ribosomes to help them perform that task for the cell. Next, we're going to talk about the cytoskeleton. And the cytoskeleton are these beams that we see crisscrossing all over the cytoplasm of the cell. The cytoskeleton is not directly involved in the making of proteins, but more the transport of proteins. Because we said that we have the rough ER, and that vesicles from the rough ER can go to the Golgi. You're seeing the Golgi right here. And how those vesicles are getting there is they're actually being transported, and they're using the cytoskeleton like highways. So here's what's going on. If we could zoom in there, there are proteins which are called motor proteins. Motor means movement. And they can interact with the cytoskeleton. So this beam is representing cytoskeleton in the cytoplasm of the cell. Inside of that vesicle, there can be molecules. So for example, if this vesicle was coming from the rough ER, we could probably assume that inside that vesicle, there would be a certain type of protein. So that protein is being transported along the cytoskeleton to the Golgi by these motor proteins. Now, I just included this diagram so that you can see that the transporting of proteins and other molecules along the cytoplasm is a pretty complex process. Next, I want to discuss a little bit more about ribosomes, because we all know that ribosomes make proteins. But how are ribosomes themselves made? Now, on a diagram of a cell, when you take a look at the nucleus, you probably recognize that oftentimes inside the nucleus there's a dark structure, a dark circle. And this is the nucleolus. this is the area where ribosomes are made. So ribosomes are actually made inside the nucleus, and you can see that over here. Now, there are two parts to a ribosome. There are two ribosomal subunits. You can see them here and here. There's a larger subunit and there's a smaller subunit. Now, when you take a look at what those subunits are made of, they're actually made of two parts. Within those subunits, you're going to find some protein, and you're also going to find a nucleic acid, rRNA. And this stands for ribosomal RNA. So according to this diagram, you can see that in the nucleolus, rRNA is being synthesized and proteins are being synthesized. And together they're going to come out of the nucleus into the cytoplasm, and they're going to be assembled into a ribosome. So to summarize, you need to be able to tell me that ribosomes are made in the nucleolus, or that's the purpose of the nucleolus, and that ribosomes are made of two types of molecules. They're made of protein and ribosomal RNA. Now we're going to get into processes other than those related to protein synthesis. So let's talk about the smoothie R, and you were taught that the smoothie R makes lipids or fats. Now, just like the Refuge sends proteins to the Golgi, the smoothie R is going to make lipids and can send them to the Golgi to be modified as well. So the lipids that we've talked about things like cholesterol and estrogen. Estrogen was a signaling hormone. So after these molecules are made by the smoothie R is going to send them to the Golgi. The Golgi can tag them for their final destinations. So the Golgi is going to tag that cholesterol to be embedded in the membrane, because cholesterol is also found with the phospholipids in the membrane. Whereas a signaling molecule like estrogen, if that cell is making estrogen, that estrogen is being made to be released because it's a hormone. So this is going to end up being secreted from the cell, because hormones will be made by one cell, released from that cell, and they'll travel to other cells and signal other cells to respond in a certain way. Now I want to talk about interactions between organelles related to energy. We had a unit in pre-AP where we talked about photosynthesis and respiration, and we know that photosynthesis is performed by the organelle called the chloroplast, whereas respiration is done by the mitochondria. Make sure you know the equations for respiration and photosynthesis. I'm going to go ahead and balance them in case you ever have to write a balanced chemical equation for photosynthesis respiration. Remember, glucose is C6H12O6. So when we go to balance this, if you just remember, you need to add sixes. It's 6 CO2, and that's for the 6 carbons here. We have 6 water molecules. There's no such thing as 6 light. So we just need to add another 6 here to the oxygen, and that's a balanced equation. When we come down to respiration, again, it's C6H12O6. So let this remind you that you need to have 6 oxygens. You have 6 CO2s that are being made and 6 H2O molecules. It's only 1 ATP being made, though. In general, in photosynthesis, the chloroplast is taking CO2 water and it's using light energy to make oxygen and sugars like glucose and other organic molecules as well. One thing I want to point out is these sugars are a form of chemical energy. So let's write chemical energy. But I want you to write unusable chemical energy. There's energy in sugars, but that energy cannot be accessed directly by reactions. So when reactions need energy, that energy has to come from an ATP molecule, not directly from a sugar molecule. It's energy, but it's in the wrong form. Whereas your ATP that is being produced through respiration, that is usable chemical energy. To give you an example, there's energy in coal, but you can't plug a lamp into it. We have to convert that energy from coal into a usable form, into electricity, and then you can use that energy to power your lamp. So in respiration, we take this unusable chemical energy, these sugars, and we can convert them into usable chemical energy, into ATP. In addition, we also make CO2 and water, and so that we say that the chloroplast and the mitochondria are interdependent on one another. What one makes, the other one takes. The products of the reactions of one become the reactants for the other process. Now in this area of our notes, I'm going to teach you some additional information that you need to know about the mitochondria and the chloroplast. We're going to have a unit completely dedicated to these two organelles. That's a very difficult unit. So if I can show you a few bits of information here, then it's less for you to remember when we're doing next unit. One thing to point out, and as you know from the endosymbiotic theory, that both the mitochondria and the chloroplast have two membranes, because when they were prokaryotic cells, they got engulfed, and so then they would have a second membrane because they were engulfed through endocytosis. Now, we can see that we have an outer membrane. Let's label that outer membrane. And then this membrane here would be considered the inner membrane. And the important thing to note is that we've created this space here that we can put molecules. And this is called the inner membrane space. Now you all know that the mitochondria makes ATP. And the ATP is going to be made right here by that molecule. So let's show ATPs getting made. And remember, it's made from ADP and attaching a phosphate onto it. But how this molecule can make ATP is because the mitochondria is going to concentrate protons in this inner membrane space. So what I want you to do is put a large amount of these protons, or these hydrogen ions, in that inner membrane space. And just like water is able to flow through a dam and provide energy so we can make electricity, these H+ ions flow through this molecule. And that provides energy for the ADP and the phosphate to be brought together to form the ATP molecule. And by the way, this molecule right here that you see here, it is an enzyme. It is called ATP synthase. So if you're asked to talk about a protein, this is always an easy one for students to remember. ATP synthase is the protein, is the enzyme that can synthesize ATP inside of the mitochondria. So let's add, the double membrane allows for the compartmentalization and the concentration of proteins. Now you can see here that we have some folds. You see them right here in that inner membrane. We know that folds increase surface areas where reactions can take place, and specifically the folds in the mitochondria called the cristae. Now going over here to the chloroplast, we see there's a double membrane. We also see that we have these stacks of green circles in here, and each circle is called a thylakoid. And I tell students to remember thylakoids instead of thylakoid. Thylakoids, let's add thyla. Because each circle looks like a coin. So one circle is called a thylakoid. Now ultimately we're going to see the same thing happen in the chloroplast. We're going to get protons concentrated, but they're concentrated inside of the thylakoid. But ultimately what's going to happen is they're going to flow through this molecule, and again, that molecule is called ATP synthase, and that allows the chloroplast to make ATP. Now I know what you're thinking. In your pre-AP biology class, you were taught that the mitochondria makes ATP, but the chloroplast makes sugars. But I just showed you a diagram, and you can clearly see that the chloroplast is making the ATP. One thing I want you to know is that ATP that is made by the chloroplast is going to be used itself by the chloroplast. So the chloroplast is not making ATP for the other cell parts to use. Whereas this ATP molecule that is being made is going to get transported out of the mitochondria, and it can be used by other parts of the cell. So if a reaction is taking place in the cytoplasm and ATP is required to provide energy for this reaction to take place, then that ATP molecule came from the mitochondria. So we were kind of just giving you a half-truth because we wanted you to answer on the EOC that the mitochondria produces ATP for the additional cell parts to use. The chloroplast makes ATP, but it itself is going to use that ATP to make sugars. Now you learned in your previous classes that cells contain vacuoles and they can store materials like water, but there's also some other vacuoles you need to be familiar with. In some single-cell organisms like paramecium are protists, and they swim around in watery environments, and many times, compared to the environment, the cell itself is hypertonic. And if you remember, water moves from a hypotonic solution into a hypertonic solution. So what is constantly happening to these cells is that water is moving into their cytoplasm. Well, if too much water would move in, then that would cause the cell membrane to rupture as the cell fills up with water. So these cells have contractile vacuoles, and you can see one right here. And this eventually fills up with water, and then it contracts, and it will then expel water outside of the cell, so the cell doesn't become too full of water to the point where it's going to rupture. We also see food vacuoles. We see waste vacuoles. And again, that has to do with single-cell organisms like this paramecium over here. And this is the mouth, and we can see that it's swallowed some food, and so that food is being digested, and eventually this vacuole is going to release the waste from the digesting of that food. Now, a couple more terms just to throw around there in case you experience them is if you see pinocytosis, that simply means the ingestion or the taking in of liquid. So there's particles dissolved in a liquid. If you see phagocytosis, then that means that we're forming a vacuole and it's ingestion of something that's solid. So when a white blood cell ingests a bacteria cell, then that would be an example of phagocytosis. Now you know that plants have a large central vacuole, and you know it's for storing water and other materials, but also I want you to remember that because there is less volume of cytoplasm, then this large vacuole allows the cells to have a larger surface to volume ratio. If you think about it, with that vacuole taking up a large area of the cytoplasm, the volume or the contents of the cell has been decreased. The surface area hasn't been changed. So if you decrease this bottom number, the denominator, that's going to end up making this number over here a greater value. So we say by decreasing the volume of the cell, we end up with a higher surface to volume ratio. Now we've already talked about the lysosomes, and they contain hydrolytic enzymes that can break down molecules. And this is using those terms that I just talked about on the previous page. Here we have a cell, and that cell is taking in a solid, and it's being wrapped in a vacuole. And we can see that ultimately it's being sent here to a lysosome, where the lysosome can break down that molecule. We also see that the cell is taking in molecules that are dissolved. So it's taking in a liquid with dissolved particles in it, and eventually that vacuole containing those molecules can merge with this lysosome, and the lysosome hydrolytic enzymes would break down those molecules. So lysosomes can digest liquids / solutes that have been engulfed, that's pinocytosis, and solids that have been engulfed through phagocytosis. We also know that lysosomes can digest old, worn-out cell parts. So when mitochondria are no longer functioning, they'll be digested by a lysosome. We've already talked a little bit about the endomembrane system because we know there are organelles, like these listed here, that are part of what's called the endomembrane system. And what we mean by that is they are all made of a phospholipid bilayer. And because of this, they can exchange materials through vesicle transport. We've already seen diagrams of the rough ER forming vesicles, and those vesicles then can move to and fuse with the Golgi and become part of the Golgi. So that's what we mean by vesicle transport. We also discussed how the members of the endomembrane system are thought to arise in cells. It's because of enfoldings of the cell membrane or the plasma membrane. And if you remember, the plasma membrane is made of a phospholipid bilayer, and so are all these other parts of the cell that are part of the endomembrane system. Now let's mention the cell parts that are not part of the endomembrane system. That would include the mitochondria. If you think about it, we never see an image of a vesicle coming from the mitochondria and moving to another cell part. Same with the chloroplast. The cytoskeleton is not made of a phospholipid bilayer. It's made of different types of protein. And ribosomes. Ribosomes aren't made of a phospholipid bilayer. Ribosomes are made of ribosomal RNA with some protein. So all those structures would not be considered part of the endomembrane system. One thing you should be able to do is tell me which parts of the cell are part of the endomembrane system and why they're considered to be part of that system, and also be able to describe to me why the mitochondria, the chloroplast, the cytoskeleton would not be part of the endomembrane system. Now let's talk more about the cytoskeleton. And when you look at a diagram, you're going to see the cytoskeleton portrayed as these beams that crisscross around in the cytoplasm. What can be confusing is sometimes those lines are not labeled as cytoskeleton, but they might be labeled as microfilaments or even actin filaments. Sometimes they might be labeled intermediate filaments or microtubules, because those are the three types of cytoskeleton. Now, it's not really important that you can list those for me. You just need to know that there's three types of cytoskeleton. The difference in these three types are their size. So the microfilaments, which can also be called the actin filaments, they're the thinnest, whereas the microtubules are like tubes, they're the thickest. And then the intermediate filaments are in the middle. You can kind of see that here. They're trying to show you that these are the thickest, these are very thin, and there's your intermediate filaments. To help you maybe remember that the actin filaments are the thinnest is I tell students to remember actin. Actin, just change it to actin. It's the thinnest of the different types of cytoskeleton. Now, here's what you need to remember about the cytoskeleton. We have already shown you that motor proteins can interact with cytoskeleton, and they can move vesicles that are containing proteins and lipids to different areas of the cell. These diagrams down here are just showing you that we have motor proteins, and they are interacting with the cytoskeleton, and they're moving vesicles that contain molecules. And these are just different ways to portray the cytoskeleton being used for transports of molecules. On the next page, I'm going to show you some diagrams related to 2, 3, and 4. We know that some single-cell protists, single-cell organisms, are able to move because there's motor proteins in their cytoplasm that are interacting with the cytoskeleton. We also know that the reason why cilia and flagella can move is because of motor proteins interacting with the cytoskeleton. And I'm also going to show you that because of the interaction of motor proteins and cytoskeleton, the cilia is able to move substances around inside a multicellular organism. So let's take a look at these diagrams. On this page, I'm just attempting to show you how cytoskeleton and motor proteins are able to move some single-cell organisms around. So this is an example of an amoeba. You don't necessarily need to explain how an amoeba is able to move, but this is what I mean by a pseudopod, is they extend part of their cell membrane out, and this occurs because of the interaction of motor proteins and cytoskeleton. And you can see them right here. So we have things like the microtubules right here, and then we also have these motor proteins, which they're represented by these little things that look like cherries. And so it's the interaction of those two molecules that are able to allow this amoeba to move around. Again, you don't have to be able to explain to me how motor proteins and cytoskeleton interact to make flagella and cilia move, but you need to just be able to tell me that that's the reason why they're able to move. If we zoom in to the flagella of a sperm cell, then what we're going to see is we're going to see rows of cytoskeleton. So this would be the cytoskeleton. And it's even diagrammed as these long green beams right here. So that's the cytoskeleton that's in the cilia or the flagella. And then we can see that these molecules, those are the motor proteins. In addition, you can also see them being diagrammed right here. So it's because of the interaction of motor proteins and cytoskeleton that the cilia and the flagella are able to whip back and forth. And then I mentioned that cilia aid in the movement of substances. One thing that the cilia assist with is the flow of mucus out of the lungs. So again, it's really the interaction of the motor proteins and the cytoskeleton inside of the cilia that's allowing for this to happen in a multicellular organism. Cilia also aid in the movement of eggs down the fallopian tube. So the cilia will move, and that propels the egg down to the uterus. And also, this movement of the cilia is allowing for these protists, which are called ciliates, which again, you don't have to know that, but that allows them to get food. So when there's food in their environment, then the cilia will move, and that draws the food into their mouths, where ultimately it's going to end up in a vacuole where it's going to be digested. Now we're still talking about cytoskeleton, but we're also going to talk about centrioles, because the centrioles are structures that are going to aid in cell division, and they themselves are made of cytoskeleton. In particular, they're made of microtubules. When you zoom in on centrioles, like you see here, so here's two centrioles, you can see that they're made of these long tubes, and each one of those tubes is considered a microtubule. We'll talk way more about the centrioles when we're talking about mitosis and meiosis. Now let me say, I'm not going to make a big emphasis about knowing peroxisomes, but I just want you to be exposed to them in case you are reading about them on an AP question. Now, one thing I want to point out in this diagram is that this diagram is just showing you processes related to respiration. So to emphasize that, let me just show you that in this process right here, an ATP molecule can be made. Now, we know that we can get energy, or we can make ATP from glucose. We can also make it from fatty acids, which you see here. But one thing that I want you to know is that peroxisomes can help break down fats and amino acids so that we can use them to make ATP. Now, something else that you might read about with peroxisomes and why they're named peroxisomes is because they have something to do with hydrogen peroxide. Whenever the peroxisomes are breaking down fats and proteins to ultimately lead to something that we can make ATP from, we get this toxic byproduct that's produced, and it's hydrogen peroxide. And it's abbreviated H2O2. Now, inside of the peroxisome, there's an enzyme which is called peroxidase. Peroxidase has the ability to take hydrogen peroxide, which is toxic, and break it down into simple water molecules and oxygen, both of which are non-toxic. So again, peroxisomes are important in the breaking down of fats and proteins so that ultimately we can use them for energy. In addition, it's able to break down the toxic hydrogen peroxide that can occur as a byproduct of the breaking down of fats and proteins. On this page, I just included some diagrams. They're very complex. Again, you don't really need to have this memorized by any means, but this is more what you need to remember about peroxisomes. And you can see it's a very complex process by which we can break down fats and proteins to utilize for energy production. We've discussed cell walls in the biochemistry unit. We know that bacteria and plant and fungi have cell walls made of different substances. So we know that cell walls offer protection. So let's indicate that to add protection. You're less likely to crush the cell that has a cell wall. In addition, I want to note that cell walls can also be semi-permeable barriers. Allowing some molecules to move through them and some things can't. Now, I know that we've said that the cell membrane primarily regulates what moves in and out of the cell, and that's true, but if you ever see any information that says that the wall is also a semi-permeable barrier, I just want you to know that that is correct information as well. So cell walls are important in protection. In plants, they also allow plant cells to develop what is called turgor pressure. We know that there is a cell wall that is around the membrane of a plant cell. And whenever we water a plant, then water moves into the plant cell. As the plant cell fills up with water, the cell membrane is going to expand, and eventually it's going to hit that cell wall, and it can't expand past the cell wall. And this pressure of the cell membrane on that cell wall is called turgor pressure. It's important that plant cells maintain high turgor pressure. When turgor pressure is high in cells, then cells are very rigid. They're very tight. And when that's the case, plants can stand upright. When we have a cell that has low turgor pressure, because it's lacking water, then those cells are a little bit squishy, and so a plant is not going to be able to maintain an upright position. In addition, we can use plant cell walls to identify the organism that we might be studying. We can look for peptidoglycan in bacteria cells, chitin in fungi cells, and cellulose in plant cells. I'm not going to spend much time on this diagram, but note you need to be able to recognize cell structures and be able to label them, so you should be able to label the majority of them. You will never have to be able to tell me the difference between a microfilament, microtubule, or an intermediate filament, but just note that those are all different types of cytoskeleton. So if you want to note that. In addition, I want to point out that these two structures are technically called the centrioles. Now you see that they're labeled the centrosome. I want you to think of the centrosome as just being the area of the cell. So let's kind of denote that there. The centrosome is really the area of the cell where the centrioles are located. I want to clarify this term with chromatin. Chromatin is just DNA when it's not coiled up into chromosomes. So when you see DNA and it's pictured as a long string, then that is DNA in chromatin form. When it coils up and it forms those structures that look like X's, then we have DNA in chromosome form. Now moving down to look at the plant cell, it looks like there's less cell parts to have memorized. I do want to note that there's some cell parts that you're never going to find in plant cells. So let's state that plant cells lack lysosomes, centrioles, and you'll never find a plant cell with cilia or flagella on it because plant cells are non-motile. They cannot move on their own. You can go ahead and cross through an amelioplast. I'm not going to make you know that. I do want you to add some cytoskeleton because I don't want you to think that plants lack cytoskeleton because you don't see it in this diagram. And then let's go over here to the underneath part of the animal cell. And let's note that animal cells lack chloroplast and cell walls. They're going to have vacuoles, but they're going to have small vacuoles, whereas that plant cell has a large central vacuole that you see there. The first thing I want you to do on this page is cross through this because all three of these cells being diagrammed here are not protist. As a matter of fact, only this one is a protist. This one is a protist. And this is a fungal cell. I want to emphasize on this page, though, that when you see single cell organisms, do not automatically think that you're looking at bacteria cells. All of these are eukaryotic cells, just like plants and animals. When you look at the diagrams, you can see that they have a clear nucleus with DNA inside of it. And then you can see the list of organelles. Remember, the definition of eukaryotic cells is they have membrane-bound organelles that can assist in functions. So again, when you see single cells, you need to note whether you're looking at bacteria cells that are prokaryotic or potentially eukaryotic cells, such as protist or a certain type of fungus like a yeast cell. Now, I just finished up showing you a typical animal cell, a typical plant cell, but you need to understand that not all animal cells are going to look like that nice round circle. Animal cells and plant cells are going to come in different shapes and different sizes. They're going to be specialized to perform a certain task for the organism. So multicellular organisms have specialized cells to perform different tasks for the organism. To get these specialized cells, we start out with stem cells. These are unspecialized, undifferentiated cells. And when they receive specific signals, that'll cause stem cells to turn into these different types of cells that end up making up a multicellular organism. So stem cells differentiate into a specialized cell of the organism. Some stem cells are going to become red blood cells in animals, or white blood cells, or muscle cells, or nervous tissue, or beta cells used to make insulin. But one thing I want to point out is that all the cells in your body, regardless of what they look like, they're going to have the same DNA. They're going to have the same genetic code. Now you're probably wondering is, how can a muscle cell and a skin cell look totally different, but yet they still have the exact same genetic code inside of them? It's because that in certain cells, some genes are going to be turned on and other ones are going to be turned off differently than in the other cells of the body. Another way to say that is the genes that are turned on in your white blood cells are not going to be the exact same genes that are turned on in your muscle cells. So the turning on and turning off of different genes in the DNA of these different cells is going to cause them to develop differently and have special characteristics that are going to allow them to perform a specific task for you. Animal cells are not the only type of cells that can differentiate. Even plants have stem cells, and these stem cells, or these undifferentiated cells, are going to receive signals to cause them to turn into or become specialized to form leaf cells and these water transport cells and root cells. And you can see that they look different because they're going to perform different tasks for the organism. So it's the differences in these structures and shapes of the cell which is going to allow them to perform these different tasks for an organism. Let's take a look at three types of specialized plant cells: a leaf cell, a water transport cell, and this is what root cells would look like. Again, they're going to be specialized. They're going to have unique features which are going to allow them to perform a specific task for plants. Leaf cells are going to be specialized to do photosynthesis, so they're going to have chloroplast. In comparison, when you look at these transport cells that are inside of this stem, so they're not going to be receiving sunlight, they would not have chloroplast. Same with root cells. They're not going to receive any light because they're underground, so no chloroplast. Since leaves are exposed to wind and sunlight, those things can accelerate water loss. And so some plant cells, leaf cells, have the ability to make this waxy material. It's called a cuticle. And that's going to help prevent water loss. So they have that unique ability to do that. When we look at the water transport cell, obviously it looks way different than your typical plant cell that you are used to seeing. They are long, dead, and hollow, so that allows area for water to travel. They have to be long and skinny so that water can move through capillary action, through adhesion and cohesion. When we look at root cells, normally they're not these perfectly square cells. They have foldings in their membranes, and that's to increase the surface to volume ratio, so there's more surface to take in things like nitrogen and phosphorus and water. Plants don't take in nitrogen and phosphorus and water through their leaf cells, so they wouldn't need an increase in surface area to do that. Now, let's look at some specialized animal cells, and I tell students to pick a couple of ones that are easy to remember and be able to discuss a specialized cell in an animal and what that cell is specialized to do and what unique features it possesses to help it perform a certain task. Let's take a look at the cells that line your intestines. Now, cells that line are called epithelial cells, and we know that food is absorbed by your intestinal cells. Well, epithelial cells are going to have a unique feature to help them absorb as many nutrients as possible. We can see that the cell membrane isn't flat here, it doesn't go straight across, it's folded. And it's folded to increase, again, the surface area in which nutrients can be absorbed. A sperm cell, that's an easy specialized cell to remember. It's going to have unique features so that it can perform a specific task for the organism. Sperm cells have to move over large distances, so it's going to be a small cell. It's going to have flagella. It's the only type of animal cell that has a flagella. Since it has to travel, it needs to be lightweight. It's going to lack many organelles. If you look at this diagram, you're going to see no smooth ER, no rough ER. But it is going to have an excess of mitochondria. If you remember, we said that the flagella is made mostly of cytoskeleton and motor proteins. And for them to interact together to make the flagella move, it's going to take a lot of ATP. So you can see the benefit of a lot of mitochondria in a sperm cell. And also, if you look at this head part of the sperm cell, you can see that there's only room basically for DNA, half of the chromosomes of the species. When we look at the lungs, the cells that line the lungs, the epithelial cells, they're going to be one of very few cells that actually have cilia. So they're ciliated. And so these cilia move, again, through cytoskeleton and motor protein interaction. And what that does is it will move this mucus up and out of the lungs. So this is a way that you're able to remove the debris that you breathe in, the bacteria that can get in your lungs. It gets stuck to the mucus. And then as these cells move the cilia, it moves that mucus and those stuck particles up into your throat region. Muscle cells are specialized to contract, so an organism can move. If we need to contract or shorten a muscle cell, then one feature is it's going to be long to start with. So we have a long object that we can contract. Something unique about muscle cells, they are multinucleic. So this is one cell right here, but these are all nuclei. And when you look in the nuclei, they're going to have the full set of chromosomes. So let's put 46 because that is the human diploid number of chromosomes. Now, the reason why it needs to be multinucleic is, if you remember, it's proteins inside the muscle that are contracting. So muscle cells have to be able to build a large amount of protein. Well, DNA is the code for these proteins, like myosin. And to make myosin, we have to transcribe that gene, transcribe the DNA. Well, that can only happen so fast. So if you only have one gene that can be getting transcribed and translated, then you're not going to be able to make that particular protein very quickly. If there's multiple myosin genes, because there's multiple nucleuses, then all of those can be getting transcribed and translated. And so this cell can make a large amount of protein in a small period of time. Red blood cells are an easy type of specialized cell to remember. They are going to be small and round so they can fit through your blood vessels. They're hollow. There is no nucleus inside of your red blood cells. There's no organelles. So this allows room for an abundance for hundreds of thousands of molecules of hemoglobin. Remember, that's a protein. And each can bind to oxygen and deliver the oxygen the cells in need. White blood cells, again, easy type of cell to remember. They're specialized for defense. We talked about in pre-AP that they're going to contain many lysosomes. Lysosomes are the organelles that will break up particles inside of a cell. The job of the white blood cell is to go around and swallow or engulf foreign cells like bacteria cells. And then those bacteria cells are going to get digested by the lysosomes. So we see an abundance of lysosomes in your white blood cells. White blood cells can also move, but they don't have cilia or flagella to help them move. They're going to use their cytoskeleton and motor proteins, and they're going to form those pseudopods, similar to how amoebas move around. Again, so pseudopods is just really like an extension of their cell membrane that kind of reaches out, and then the rest of the cytoplasm kind of flows into that area. That's what we mean by pseudopods. On this page, we're going to briefly revisit some ideas related to those 4 big ideas of AP biology, in particular the 4th one, that biological systems interact, and these systems and their interactions have complex properties. And again, we're going to talk about this idea about robustness. It says that when a biological system possesses many different varied components, like differentiated cells, different cells, it's going to have greater flexibility to respond to changes in the environment. And again, this phenomenon is called robustness. So we're going to just emphasize that variation in cells, you have muscle cells, red blood cells, white blood cells, is going to provide you with a wider range of function in comparison to this little bacteria cell right here. So let's think about this. Let's say that you walk out into the sun and you have bacteria on your skin because you are covered in bacteria. Now, if you stay outside long enough, then this sun is going to kill a large amount of bacteria cells that are on your skin. But yet the sun doesn't kill you. Well, it could, but it doesn't. Why? Because you have so many different specialized cells that are going to help detect stimuli and allow you to respond to the sun, so you're not going to get damaged or hurt by the sun. So because you have a variety of cells, then you're going to be able to better respond to changes in the environment. You're going to be more robust because of your differentiated cells compared to this little bacteria cell that is just one single cell. You're going to use your muscle cells to move into the light. You're going to have sweat gland cells that are going to produce sweat to keep you cool. Your skin cells are going to produce melanin, and that's going to help protect your DNA from the UV. I can go on and on. But again, you're going to have a greater chance of surviving. You are more robust because you have variation in your cells that are going to help you respond to a change in the environment. And then again, another idea related to big idea 4 is that biological systems have parts that interact. These parts will form new structures, and these new structures will have properties that are different from the individual parts. So let's talk about what's called the level of organization, from simplest structures to most complex. You ultimately are made of atoms, like carbon, hydrogen, oxygen, nitrogen atoms, and they come together to form molecules like proteins, carbs, lipids, and nucleic acids. These molecules make up organelles. I'm going to put organelles up there. Organelles make up cells. Cells make up tissues, and there's 4 different types of tissues. There's epithelial. Again, those are cells that line parts of the body. Like the lining of your stomach is considered epithelial tissue or epithelial cells. Connective. So there's cells that come together to make connective tissue, like tendons, and blood is considered connective tissue. There's muscle tissue. And there's nervous tissue. So those are the 4 types of tissues. Now, it takes these 4 types of tissues to make up organs. So every organ is made of these 4 different types of tissues. So in the stomach, we can find epithelial tissue, connective tissue, there's muscle tissue in your stomach, and there's nerves that are part of your stomach. Organs come together to make up organ systems. So along with the stomach, we would have the pancreas and the small intestines and the large intestines, etc., making up the digestive system. And then finally, it takes many different organ systems to make up the entire organism. So let's talk about this idea that the individual parts are going to have properties that are different from the properties of the whole. Let's discuss organelles, since we just talked about all the different organelles found in cells. Well, one emergent property that results due to the interaction of these organelles is cells are living. And I had that for you right here. So this is a property of cells, but you cannot apply that property to organelles. The smooth ER is not considered living. Neither is the mitochondria or the chloroplast or the nucleus. So this property emerges just due to the interactions of these individual cell parts or these organelles. Now we're going to go back to the very first page of this note section and go through these blanks to make sure you have the correct answers. The plasma membrane, we talked about, is made of phospholipids and proteins. It regulates what goes in and out of the cell. The arrangement of the molecules in that plasma membrane, like the cholesterol, the marker proteins, and the phospholipids, that model is called the fluid mosaic model. We believe that many infoldings of the cell membrane led to those cell parts of the endomembrane system. We know that it contains this cholesterol, which is a steroid. This acts like a structural buffer. So what we mean by that is whenever temperatures get too warm, then the cell membrane is not going to become too liquidy. Whenever it gets colder, the cell membrane is not going to become too rigid. So it's not a temperature buffer, it's a structural buffer. The cell wall, we know that in plants it helps develop turgor pressure. It offers protection to cells. In plant cells, we learned it's made of cellulose, chitin in fungi, peptidoglycan in bacteria cells. The smoothie R makes lipids. I tell students to remember that when you want to make your hair smooth or your skin smooth, we rub fat on it. Conditioner is just fat, it's oils. Same with lotions. If you look at label on lotions, it'll say like palm oil, coconut oil, and so on. So you're rubbing fat on your hair and on your skin to make it smooth. So try to remember smooth going with fats. As for the ReFER, we know it's covered with ribosomes. It's going to modify proteins. From there, it's going to send them to the Golgi. It is one of many organelles that are part of the endomembrane system, again, due to the infolding of cell membranes of early prokaryotic cells. The mitochondria makes the ATP through the process of respiration. This requires oxygen and glucose. The mitochondria has two membranes. The inner membrane is folded, and this is to increase surface area. We can have those ATP synthase molecules for the ATP production. It has its own DNA, and it's circular, just like bacteria DNA. It has its own ribosomes. We find an abundance in cells with high energy demand, like muscle cells. We just talked about sperm cells. The chloroplast does photosynthesis. In photosynthesis, it's producing organic molecules. Many of you probably put glucose there, but photosynthesis can produce other molecules besides glucose, but primarily it does produce glucose. It has that green pigment called chlorophyll. Chlorophyll is found in those thylakoids. The Golgi is the shipping center, and what's being shipped to it would be proteins from the ReFER, lipids or fats from the smoothie R. Golgi is going to tag them, modify them, send them to their final destination. This is also the source of lysosomes. Lysosomes are made by the Golgi. The nucleus contains the DNA, which are the blueprints for building proteins. To fit all that DNA, like 6 feet of it inside of a nucleus, is wrapped around proteins. Those proteins are called histones. When DNA is wrapped up around these histones, then it forms chromosomes, but uncondensed DNA, remember, is called chromatin. We talked about chromatin coiling up to form chromosomes before cell division when we talked about mitosis and meiosis. Now, attached ribosomes are going to make proteins that can be secreted or exported from the cell. So these proteins are going to be sent to the membrane by the Golgi, and then they're going to be released into the environment of the cell. Some of these proteins, though, are going to become embedded in the cell because they were transport proteins or marker proteins or receptor proteins. And some of these proteins are going to end up being the hydrolytic enzymes that are inside lysosomes. So after these proteins are made by the ribosomes, sent to the ReFER, ReFER is going to send it to Golgi, and then pieces of the Golgi with these enzymes inside of it are going to break off. Because remember, lysosomes form from the Golgi. Your unattached ribosomes that are floating around the cytoplasm are going to make proteins as well, but they're going to stay in the cytoplasm for the cell to use. And we're going to talk about specific enzymes that are in the cytoplasm. They're going to help with specific reactions. You're going to be able to name them here, like kinases. They also can make proteins for other organelles to use. We're going to find many more ribosomes in cells that have to make a lot of proteins, like those muscle cells. And if you remember, ribosomes are found in all cell types. So this is something that's common to all cells, and it indicates the common ancestor of all living things. Do remember, though, that the ribosomes are smaller in prokaryotes. Eukaryotic cells are going to have larger ribosomes than in bacteria cells. Moving into the next page here. We mentioned that we find abundance of lysosomes in white blood cells. White blood cells are going to be engulfing bacteria and breaking them down. Vacuoles, we find them in plant cells and animal cells. They're larger in plant cells, mostly to store an abundance of water and help plants establish that turgor pressure needed to help those cells be tight and the plant structures to be upright. This large vacuole that we find in plant cells, it allows for a greater surface to volume ratio because it creates less volume. There's less room for organelles in the cytoplasm because of that big vacuole. And remember, it's part of that endomembrane system. The cytoskeleton, those are the beams that crisscross. They serve as highways for the movement of vesicles. The cilia, with those short hair-like structures. Flagella, or the longer tail-like structures. And that pretty much does it. So again, I've included the information that you probably learned in past classes in the first column, but we did go through some additional information as we were going through these pages below. And so in addition, you'll need to know this information as well for your assessment.